I did a fall. One time I fell down in class. That was a memorable experience for everybody. Oh, could happen here today. All right, here we go. This is our class. We're meeting now. And here we are. Welcome. And, okay. And so, what you do this week. So we'll finish talking about chapter 7 today. And we'll talk a little bit about chapter 8, and then we'll talk about chapter 8. Chapter 8 is about differentiation in stem cells, so we already have some knowledge about that. Chapter 8 is a chapter where our author talks about the clay model videos. There's a really good chance that you will just want to go back and watch all those for the second exam, because although it was material covered on our first exam, and you don't necessarily need to review the material from the first exam, it's that material. And so I decided, and this will be available when I put it up on the web, so you don't necessarily need to scramble. We're gonna cover those pages in chapter 8. So we're not gonna look at the rest of the chapter ever. Those are just the pages we're gonna cover from chapter 8. And that's just because we don't have a lot of time before our exam, which is 1 week from today. And we've known for a long time what our exam is going to cover. It's going to cover fruit fly chapter, plant chapter we're talking about today, sort of the cell biology of development, and then these parts of chapter 8, differentiation and stem cell biology. So then, neural crest migration drawing assignment is handed in today, or send me pics by email today. And then you're working on your term paper, and I think I've talked to just about everybody about that, and then talk to them about a first round of like, that's a good paper, and then a second round of, here's some highlights to help guide you to that work. And I know we're focused on the exam right now, so you need to be working on your term paper, but we'll talk especially about the exam. Okay, so chapter 7, we have this image. We wanted to talk about cytoskeleton, so we learned some features of that. Right now we're talking about the cell adhesion molecules, then we're going to talk about the extracellular matrix, then we're going to talk about examples. The one example we already have is the neural crest cells. Another example we already have is the macaroni assignment from the fruit fly chapter, because the macaroni are cell adhesion molecules. Okay? Does anybody have any questions they want to ask right now about anything at all? Okay. And you can just interrupt, because this week, today and Thursday, of course, we have to review too. So for cell adhesion molecules, go to this box for getting that introduction that we want for our class. And the author talks about 3 families of cell adhesion molecules. Remember that cell adhesion molecules are super famous in the history of life on Earth in animals. Every animal, all the way down to sponges, has lots of these cell adhesion molecules. The bigger, more complicated ones like us have lots and lots of these, and they're in gene families. So we have cadherins. Cadherins are a protein. These are all proteins that are cell adhesion molecules in the membrane. And there's a family of paralogs in us, because they're like screwdrivers. They really help and hold cells together, and they do a sort of a structural role. They also have an information role. So the cell adhesion molecules, when they hold on to something, are going to be communicating that fact into the cell. So they're going to be stimulating the cell to make a cellular response. And so here are the first of the 3 categories. It's the cadherins. We started talking about it a little bit last Thursday. Cadherins is a good name because cadherins hold hands with other cell adhesion molecules on other cells called cadherins. So holding hands, like cadherins are like a type of macaroni, and they hold hands in a calcium-dependent way. So why are we learning about cadherins? Well, first of all, they're very, very common in lots and lots of animals, lots of orthologs, and then families of cadherins in organisms like us, animals like us, and their hand holding is dynamic. When we learned about the macaroni and the fruit flies, were those cadherins? Probably not, because that's a hand holding that they're just going to hold on for the life of the little fruit fly. Cadherins are ones that can bind and let go, and just by regulating the amount of calcium. So that's the first thing we want to say about cadherins. Then here's the next thing. The next thing is you can see that when calcium is present, the cadherins are binding and holding hands, and then they also bind to actin microfilaments. And so the actin microfilaments are playing a role in the cytoskeleton of the cell, and that role can be to change shape and be a structural component. And what the cadherins do is they link the cytoskeletons between cells. So that's one way to build a stronger connection and a coordinated connection. Remember that we said, oh, you know, when morphogenesis occurs and the belt narrows and folds in, those cells are changing shape. And it's individual cells changing shape. Maybe they were a cube and now they're more of a trapezoid. They're also communicating and coordinating that cell shape change. So this is a good example of that, right? Where we have the cytoskeleton, it's really the shape of the cell, and the cadherins are connecting those. So they're both structural and informational. So that's the second thing. First thing about cadherins is calcium-dependent binding between cadherins, between cells. Then second thing, they connect the cytoskeletons between cells and build a stronger structure that could fold and bend and stuff. Third thing is there's you can see the actin microfilaments here, and then there are some connector proteins that build a bridge between the cadherin and the actin microfilament. And one of the proteins is beta-catenin. So that's the third thing we want to know about cadherins is we know what beta-catenin is. It's a transcription factor. And it has target genes and it activates target genes. And so it's going to cause changing gene expression. But it has to be in the nucleus to do that. And when there's high calcium and the cadherins are bound, they're including beta-catenin here in the cytoplasm. So what that means is that when the cadherins are bound, beta-catenin is not doing its job, and when they let go, beta-catenin goes to the nucleus. So that's a good example for us of how these dynamic cell adhesion molecules can affect the cell by changing gene expression. When cadherins let go, new gene expression occurs. The beta-catenin is released, it floats into the nucleus, binds to the regulatory region, and activates target gene expression. And that's an important concept for us, the idea of the cell adhesion molecules being structural, but also informational. We have that combination, and that combination is true for lots of cell adhesion molecules, that they play a structural role and an informational role. Yeah? And so cadherins are a great example for us to pick on. For the book to introduce, if you only could talk about one, probably talk about cadherins because they tie things together in such a pretty example as that. Okay? And then that's the first one, cadherins. And our book in the box talks a little bit more about them. We just want to have that going forward so we can use it as examples is cadherins. They're protein, cell adhesion molecules like that. Okay? The next one are called CAMs, C-A-M. And it's true that CAM is an abbreviation for cell adhesion molecules. So that's a little frustrating. But I'm pretty sure that CAMs were the first one that was discovered. So they're like, these are cell adhesion molecules. And then people are like, oh, there's lots of different ones. And so let's call those CAMs. All right? So this is an example of CAMs. CAMs are like those macaroni molecules that we learned about in the climate, in fruit-wise, across the anterior-posterior boundary compartment. And they hold hands like that, the elbow macaroni holding hands with elbow macaroni. And what we want to remember about these guys is they make really stable hand holds. Lots of times they just hold hands for the life. So if we had to guess, we might be wrong, because there are other classes of cell adhesion molecules, but we just had to guess, we'd say those macaroni molecules were probably CAMs. Here's a really good way to remember that. So the CAMs often have names like this one, and we can learn the name of that specific family member because it's family. It's called NCAM, and it stands for neural cam. And here's a good image of that idea. So it's showing the development of the neural tube in a vertebrate. And so let me just draw that, you know, to just kind of remind you where we're at. Okay, so remember when we were looking at frog development, we would draw a circle like this and we'd say, oh, there's the notochord, and we've got somites here, you know, and we've got lateral plate mesoderm here. So we got that cross section. And here's the overlying ectoderm. And the overlying ectoderm is going to be stimulated by the notochord through signal transduction factors to change shape, fold up like that, and then pinch off and form the neural tube. So what's going on is initially we have this ectoderm that's thinking that its fate is going to be the skin, the epidermis. And then this signal changes the fate of these cells, so they start to decide that, yeah, they're ectoderm, but they're going to become the neural tube. So their fate is going to be the central nervous system. So you can see right at this point in development, we have to create two teams, the eventual epidermis team and the eventual brain and spine team. And that happens because these cells, as part of that signal, they start expressing NCAM. And so these cells start to hold hands with each other, and then they're going to stay separate. So one way to segregate or separate these cells is to have them hold hands. So let's start having this team hold hands, and they'll stay together. So NCAM is a great word for us to remember. It is a stable hand-holding cell adhesion molecule that will hold hand between cells, and the development of the vertebrate central nervous system is a perfect example of that. And it's shown in your book. Remember that when my friend Sarah, the sailor, was here, I wanted to talk about sponges because she likes sponges because she sails over the ocean. But also more relevant for us is that sponges have these kind of molecules that hold together, and if you disperse a sponge and just leave it overnight, it will just put itself back together. That is seen here in this experiment, where when the neural plate is forming, so during neurulation in a frog, you could take these cells off, disperse them, they're living cells, but you separate them like that, mix them together, and then they'll separate. And they're going to separate because of NCAM. The cells bump into each other, and when the NCAM molecules bump into each other, they hold on, and you just get this natural separation. So it's a really good example for us to think about NCAMs and this idea of holding hands and holding on. Again, we don't know for sure if the macaroni molecules were CAMs because there are a bunch of different cell adhesion molecules. You know, and they have overlapping sort of roles. But that's a really good one. NCAM is just a great one for us. There are orthologs of NCAM in every animal that has a nervous system, holding those cells together, making them distinctive ectoderm. And that's what we want to say about NCAMs. Another great example, yeah, big family of those in our body, different forms of CAMs. Okay? Then the third one of these are called integrins. And here's the thing we want to see with the integrins. Great example to pick because you see, integrins do not hold hands with each other. First two examples, they're holding hands with each other. It's two cells holding on. Integrins hold hands with parts of the extracellular matrix. And that's a really important concept for us too, that when the neural crest cells are migrating, they're making connections between cell adhesion molecules and other cells or cell extracellular matrix. So we gotta learn that concept too, that integrins connect the cell with the extracellular matrix. They hold on, it could be a structural role. It's especially thought of as an information role, and that's what's shown here with this red arrow, that when integrins are holding on to that molecule in the extracellular matrix, they're sending a signal. Almost like they're a receptor, and they're like, yep, sending that message and changing the signaling pathway into the cell and causing the cell to respond. And so that's what we want to get out of integrins. Now again, there's a lot of overlap to these stories, but as an introduction, this box, one-page box, is really perfect because these are really famous examples of the cell adhesion molecules, and they play really important roles in us, and we can immediately kind of go, yep, okay, got it, you know, for each of these categories. So we'll plug some of those into specific examples, but especially, you know, on the exam, it could be fill in the blank, it could be short answer question, you know, like what are, you know, 5-point question about like what are 3 cell adhesion molecules, and, you know, describe each one, or draw a little picture and describe each one. And you'd be like, okay, cadherins and CAMs and integrins. Yep, okay, and so that's really what we're going to say about them. So that's just an introduction to them, and then we plug their roles into examples, okay? So then the third one is the extracellular matrix. And that's that one that was really important when we could decellularize an organ, and then if we repopulated it with stem cells, the stem cells could create a new organ. Just as a way to say there's just tremendous amounts of information in extracellular matrix. And it's made over time in development, so as the embryo is growing, the cells could be making different extracellular matrix components and different concentrations, and they can be establishing breadcrumb trails. And the breadcrumb trails are stuck, they're anchored in the extracellular matrix, and that's how the neural crest cells are crawling primarily, is they're following a breadcrumb trail that was laid down earlier in development when the embryo was really little, and as it got bigger, it just naturally laid down these trails. So it seems shocking to see that. And there are lots of examples. Anytime a cell is migrating in our body, it's migrating following chemical trails, direct contact trails in the extracellular matrix, okay? So this is what we want to learn about molecules in the extracellular matrix. We'll learn about 3 families of protein and then 2 polysaccharides. So the extracellular matrix is generally made of long polymers of proteins or long polymers of sugars, polysaccharides. And we're going to learn collagens, fibronectins, and laminins for proteins. And you'll see we don't have to learn very much about them, but it'll just be a list of examples. And then we're going to talk about a specific example in a few minutes of sea urchin gastrulation, and we'll talk about those 2 there then. And there are hundreds and hundreds of different examples of proteins and complex polysaccharides in the extracellular matrix. And so this is just a little introduction. And lots of times in the extracellular matrix, the proteins have sugars connected to them, covalently bound to them. Those are called glycoproteins. And you might have learned that term in introbio. So we have proteins, we have polysaccharides, sometimes we have glycoproteins. That's a protein with a polysaccharide attached to it. Sometimes the protein is pretty small and the polysaccharide is really big, and those are called proteoglycans. And so there are a couple of specialized terms that you might have heard before that fit into this category. Again, extracellular matrix, lots of different molecules, but we especially think of these long chains because they play a structural role. Our cartilage in our nose and in our other joints and so forth, the cushions in our joints, that's extracellular matrix. That's providing that cushion and also that strength and structural support, right? So you can run on your knees until your extracellular matrix cushions just get all smashed down when you're old and you have sore knees all the time, like that. Okay? So structural, yeah. But then tremendous amounts of biochemical information that's anchored there. All right? So here's just this, right? If you look up integrins, right? So we just talked about integrin, and you remember those are cell adhesion molecules that bind to components of the extracellular matrix. There are lots of different members of the integrin family, and when they're bound, they're sending a message to the cell. I bound my target. Okay? So just look at this. What are they, right? Integrins are transmembrane proteins that bind to extracellular matrix components, including collagen, fibronectin, and laminin. So lots of different members of integrins, and they bind different targets. And that's one reason why we learned those three proteins, because they're famous targets for the family of integrins. So a specific integrin family member will bind, it's supposed to bind collagen, oh, there it is, bound it, and so it's gonna send signal back to the cell, I bound it, okay? Integrins are heterodimers made up of alpha-beta subunits, each with an extracellular domain, transmembrane helix, and cytoplasmic region. Great, they're transmembrane proteins. When an integrin binds to a ligand, the target, the cytoplasmic tails interact with intercellular proteins which can affect cellular behaviors like survival, differentiation, and apoptosis. So cell responses, when the integrins are bound, they're going to cause the cell to make responses. And there's some really important responses that we've learned about, like apoptosis. If the cell isn't found its target, it could just kill itself through apoptosis. Or it could differentiate, right? So, you found your target, now you can stop and differentiate, okay? So, just an example, like why those three? Well, because they're super famous and their families, and you've heard of collagen and fibronectin and laminin are just about as famous as collagen in this field. Yep.
When it says cytoplasmic tails, is that talking about like the phospholipids?
No, it's like this, where if we drew, it says they're transmembrane proteins. So that means they span, they cross the cytoplasmic, across the cell membrane. So if we just looked at the cell membrane, right, that's the cell membrane, and it's that phospholipid bilayer. And a transmembrane protein, like integrin, crosses the membrane. And so it would have an extracellular region, and extracellular region is called a domain, so an extracellular domain. And if this is an integrin, then its extracellular domain has a shape so it could bind something like collagen. And when it's bound to collagen, it changes its shape. So you could draw it, and it might look, it says it's a heterodimer, so it might look kind of like a pair of pliers, where when it binds collagen, it actually has a shape change, and it could change from shape like this, more open like that, to like a pliers would close. And as a result of the closing, the cytoplasmic tail shape changes. So that's the concept of a extracellular protein, is that we could have an extracellular domain, and when it changes shape, that shape change is communicated into the intracellular domain. And that's the first step of that information flow. When it binds, the information is brought into the cell by the changing shape of the integrin protein. And when its shape has changed, it binds to cytoplasmic proteins that do tasks to further the message coming into the cell. Like maybe they move to phosphorylate targets and things like that. Yep. So transmembrane protein with an extracellular domain and intracellular domain. Yep. And so the cytoplasmic tails are like that part that is going to further communicate the presence or the binding of the integrin into the cell. Yep. And so that's why we picked those guys. And we'll talk a little bit about more examples, but those three protein families, and you can see there's some variety there. Let me just show you a little image of, if you look at collagen, it immediately starts talking about different family members, type 1, type 2, type 3. Those are different paralogs. And we have several collagen family members, slightly different. They probably have really similar structural characteristics, but they have a different potential for information, right? Because probably a different integrin binds to the different collagens, and that provides opportunity for better variety of messages. So not just one breadcrumb trail, but a variety of breadcrumb trails are possible. Then if you see in fibronectin, same thing, where it starts talking about different types of fibronectin. Those are different genes in the fibronectin gene family that code for slightly different fibronectin proteins. They would have slightly different targets, maybe bind a different, form of integrin. It mentions integrin here because it wants to talk immediately about why there are different family members. Here's laminin, again, a naming. Of course, we don't need to know the names, but we've learned about gene families and how the gene families are arose by random gene duplication, and then it's like helpful. And it's like a screwdriver, where you have a simple toolkit, you have one screwdriver, but it's really helpful if you have 2, 4, 6, 8. And so that expands the possibilities of these closely related proteins in this gene family. Yep. And so that's that. And with that, we're done introducing the extracellular matrix proteins. We'll loop back and talk about the polysaccharides that we want to learn about when we get to this example. So now we're just going to roll through some examples, some of which we've already covered. Okay? So there are stories about morphogenesis, about animal development and how shape is developing in animals by cell movement. And there are really 2 kind of categories of cell movement. The first is where a cell moves past its neighbors quite a ways, and that's called cell migration. And so our good example of that is the neural crest cells. They crawl past lots of cells to get from the center of your back to the face. So that's a really great example of neural crest cell migration. Anytime any animal cell crawls like that, migrates, moves past other cells, it's moving like the same way the neural crest cell is moving. This is not a course in neurobiology, but I pulled out this image. This purple is the tip of a nerve cell that could be sending a long connection, an axon, a connection from your spine all the way to your big toe. And the cell isn't moving in this case, it's just that tip that's migrating. And the tip has everything we're talking about. So cell migration is cells moving past each other, or it's a special example of where a nerve cell is growing out long distances to make connections. In that case, the nerve cell isn't migrating, it's just the tip of it. And if you know the terms, it could be an axon or dendrite. And this just shows, like, put an arrow here, laminin, fibronectin, collagen, tenacin, just extracellular matrix components, and it's showing integrins making connections. Integrins are a really good example. When we think of the neural crest cell migrating, we'd be, those are probably some integrins doing that, because they're reaching out, making those connections, and following that breadcrumb trail, and following it by providing these cues. Let me just find this slide. It might be, yeah. So remember, in our neural crest cell migration, we said the cell is moving that way. It's not sending out filopodia randomly. It's really orienting the filopodia toward that way. And it's doing that for 2 reasons. Both reasons are examples of chemotaxis, so using chemical cues to decide which way to go. And we've just been talking about what are called short range or short distance chemical cues. They're direct contact. So examples of chemotaxis and cues in neural crest cell migration would be collagen, laminin, fibronectin. They're there, and the little neural crest cell is making contact, reaching out, and there's probably a higher concentration as it's going forward, going, oh yeah, oh yeah, oh yeah. So it's sending that signal back into the cell, and that's that example. These short-range chemotactic cues are proteins in the extracellular matrix. They could also be some proteins, some of cell adhesion molecules on the surface of cells that it's crawling past, but especially significant is the extracellular matrix. And then just remember that in addition to that, there are long-range, long-distance cues, and those are signaling factors like we've learned about, where in that case, a source cell, maybe up here, is making a signaling factor, releasing it, and there's a concentration. And the neural crest cell has the receptor, and it's receiving that signal and saying good, and as the concentration increases, it's better, better, better. Kind of like that game hot, cold, you know, where you're like warm, warmer, warmer. So long distance cues can be that, going up a concentration, up gradient, or maybe going down, so trying to move away from the source of a chemical signal like that, okay? All right, so that's our example of cells that are actually moving past each other. That's one category. The other category is where the cells really aren't moving past other cells. They're really changing their shape or jostling, kind of shifting. And so we'll talk about 3 examples of that. I guess I've got these a little bit out of the order that I want to talk about them, because I was just reorganizing slides. I think I have here. Oh, and before we move on, let me just show you a few more slides I have about that neural crest cell migration. So, you know, these are just some terms that came up in that neural crest cell migration assignment, and these are ones that you might have to rewrite. You know, draw a picture of neural crest cell migration on the second exam or the final, and use all the terms, you know, explain all the terms briefly. So it'd be a drawing, label with terms, right? And you can just see, right? First exam was a little bit surprising. You know, we covered a lot of territory. Well, we're covering a lot of territory now, and you can see once we have a foundation, the territory can fall into more chunks, right? So that could just be a 20-point question on the second exam or the final. It'd be a good one. And you know, we did it as assignment, you know, so you're set up, right? And then we have the fruit fly stories, right? You can also see how these could be fill in the blank or shorter answer questions like that.
Would you give us that list?
No, I hope not. That's a complicated thing though, because like, you know, I gave you some lists on the first exam. I'd ask you a question, I'd say, make sure you use these terms. And it's hard because, you know, you'd say, what's the kind of, you know, like there should be a rubric that I use for scoring. And so I like to not give the terms to see if the student has learned what important terms there are. But when I do that, you know, then I'm starting to grade like a rubric where I'm saying, well, if you are writing about neural crest cell migration, you better say something about it, integrin. And so I'll be like, right, there's the integrin, explain, you know? But then some students won't explain it, but they'll explain everything else really well. So it's hard. And so sometimes I decide to make a list, and sometimes I don't. And it's hard because if I don't, then I'm like, ugh. You know, how did Zach not say integrin? Oh, I'm mad about that, right? Mm-hmm. But yet the rest is good, so it's hard to grade that. On the other hand, we saw that in the first exam, if I give you a list, students think it's just, oh, I'm just supposed to define these. No, you're supposed to use them in the drawing and in the explanation. Put them in context rather than just say that. Yep. Yep. And so we have some massive setups, right? Because these are examples that we've had some assignments or some examples in class, right? And we have the plant chapter 2. Yeah, okay? Okay, so just to show you this, and again, you don't have to look at this hard, but just showing this various extracellular matrix components during neural crest cell migration. Look, the component, right? Fibronectins, laminins, collagens, different types of collagens, yeah? This is just another one. And then it says, you know, is it providing a permissive? Does the cell want to crawl that way when it detects it? or non-permissive, doesn't want to turn and crawl away from that. And you can see that these proteins play that role. And it's just great because it shows you that one family can play a role, by having different specific members, and you're like, oh, I know what collagen is, right? And helps hold our body together. Well, it also provides this information in the extracellular matrix. So just a little information about that. Okay, just a visual about that. And then a visual about this. So we talked about the neural crest cell migration. And I just wanted to show you this. These are the skulls of some vertebrates. There's a human skull, and there's a mouse, and there's a bird. And we'll focus on the human. The colors are showing homologous parts of the skull. But if we just focus on the human, you probably know that in our skull there are some cracks called sutures, just little visible wiggly cracks in our skull, okay? So there's some lines like this, okay? And that's what it's following here. So see that whole front part of our skull is a product of neural crest cells. So the neural crest cells are making the vertebrate face, and it's astonishing that includes the skull in the front, because it also makes all the purple parts. Those are normally mesoderm, right? Ectoderm does not make bone. Ectoderm famously makes the epidermis, skin, and the central nervous system, but not bone. And so the reason those sutures are there for some of them is because it's a profoundly different source of cells that make the different parts of the skull. And you can kind of see here that ancestrally, prior to vertebrates, there was some head case of mesoderm derived bone, but the vertebrates are like, no, we're going to make a really complicated face. And that's like the ancestral root of the vertebrates. And then you can just think about the variety of vertebrate faces, right? From fish, salamanders, and so forth, mammals in the ocean versus mammals on land. Great diversity in the structure of our face. So people will say, vertebrates have faces. There's no invertebrate that has a face. An octopus doesn't have a real face. A fruit fly doesn't have a real face. Vertebrates have faces, and that's part of the reason why we see such tremendous diversity of vertebrates, right? A bird beak versus a mouse. Skull looks very different than ours, and so forth and so on, like that. Yeah? Okay? So then here's stuff like this. Okay? So you see that diversity, but then see these identical twins, and you probably know identical twins, that is very hard to tell them apart. So that shows that, there can be tremendous diversity within our species and between species, tremendous diversity. But when you look at identical twins, that action of the neural crest cell migration and the formation of that frontal part of the skull and other tissues in here is remarkably, the word is robust. That means it's reproducible. It just makes the same thing in the twins. Really, really similar. Yep?
I didn't have a question. Sorry.
Yep? Yes?
No, I didn't have a question.
Oh,
I was scratching my
you're
head.
just reporting? I do not have a question. Yes, say that every few
'Cause I thought you said
seconds.
yes.
I don't have a question. You may move on. Okay? So then there's cool stuff in the evolution of vertebrate face. One is the transition from primates to humans, like that. We have a different shape. They have more of a jutting jaw like that, right? So it says how we came to ourselves. So part of the evolution of humans includes a change in that facial structure. That's really interesting, because if you look at the genes that are understood to have changed, they're not new genes. They're just some mutations in genes like sonic hedgehog and other regulators of it. And they say tamed because they compare that to the difference in domestic dogs versus the wild dog ancestors, and domestic cats versus the wild cat ancestors. Because we tamed those, we domesticated those pets over thousands of years, they have changed facial features from their wild ancestors, and they include lots of the same genes. Whether it was a kind of natural selection, evolution of the human, from primates, or this artificial breeding selection of cats and dogs. So there's just all kinds of really cool examples of developmental biology and vertebrate face. And so neural crest cells, they migrate to form the face. We want to learn a couple of different roles that neural crest cells migrate to do. So one of them is neural crest cells make the face in all vertebrates. Fantastic. Here's something we haven't talked about that's in chapter 7, and so we'll just learn it to increase our list. The second thing is neural crest cells also migrate to form what's called the peripheral nervous system. So we have a central nervous system, the spine and brain, and we also have a peripheral nervous system. And our spine and brain are used for thinking and also for controlling muscles. The peripheral nervous system is used for sensation. And we have our organs, that's part of the nervous system. When we say peripheral nervous system here, we're really focused on the sense of touch and also the sense of kinesthesia. And kinesthesia is how you know where your body is in space. So you know how you can close your eyes and touch your fingers together. That's because you know where your limbs are in space. And that's the part of the peripheral nervous system that we're focused on here. The neural crest cells migrate to form the peripheral nervous system. And we'll talk a little bit about that because our book has a figure of that. And then we'll just mention this last one because that's 3 really famous roles that migration and neural crest cells play in all vertebrates. Is form the face, form the peripheral nervous system, touch and kinesthesia, and then they also form all the pigment cells in all vertebrates. Okay, so here's a figure from your book. We're looking down at any vertebrate, and these are showing, that's the neural tube, and then these lumps are showing somites. And so, yeah, I'm just gonna, another non-question, yep, good. And I'm just gonna redraw this, and we'll look down, so we're looking down, like a vertebrate is laying on its tummy, and we're looking down at it. And so here's the neural tube. And then, yep, and so that's gonna be the neural tube, and that would be the brain up there. So we're just looking at this portion of the neural tube, and then these are gonna be the neural crest cells, so they are right there, and they haven't taken off to migrate yet. So those are gonna be neural crest cells. And it's the neural tube. And then the drawing, the figure just has 2 pairs of somites. So there are 2 pairs of somites. And some of these neural crest cells are going to migrate, and they're going to form the peripheral nervous system. And we have our sense of touch out here. The cells that we use to feel, like that, whether it's the sense of touch or this kinesthesia, the cells, the nerve cells that are doing that, those nerve cells are actually here. So when the neural crest cells migrate to form the peripheral nervous system, they don't move very far. They do migrate, they crawl past other cells, but they don't move very far. They move to make clusters of cells here and here like this, and on, up and down the neural tube. And so we get to show the neural crest cells, and these neural crest cells will crawl, and they'll form this one, and then these will form that one. So they crawl just a little ways to make these clusters, and the clusters develop into what's called dorsal root ganglia. And so these are little clusters of nerve cells that are up and down our spine on either side. And we're just gonna overlay our vertebrae. So this is where, let's see if I can get a good color for that. So this is where the vertebrae, the bones of our backbone, that's where the vertebrae are gonna be. So you see the dorsal root ganglia, I don't know, I should probably make that narrower. The dorsal root ganglia are in between and flanking our vertebrae. Okay? And so they migrated, but not very far. And that gives you, the landmarks to see this. So what it's showing here is, this will be, that's just one somite. And it's showing that, there it's labeled, I didn't see it before. So the somite is labeled, it's actually, let me erase that because, get this right. So, because this is the actual somite here. And what you can see is the somite is going to have an anterior yellow region and a posterior gray region, each somite. And what you can see is that the neural crest cells don't like the gray part, they like the yellow part. And so when they migrate to the anterior region of each somite. And it's because the cell adhesion molecules are different on each somite. So each somite is made of cells, mesoderm cells, that are going to develop to be bones and muscles and so forth. But early on, they're just somites, mesoderm, and they have different cell adhesion molecules. The yellow could easily be described as the elbow macaroni, and the gray, the shell macaroni. And the neural crest cells only want to crawl on the elbow macaroni. And that's how we get the smooth row of neural crest cells to migrate to form these spots, these individual ganglia. And so there's the dorsal root ganglia, that sort of dark orange and white. And the dark orange and white is going to send out connections to the tips of all our fingers and toes and into our muscles, because our muscle and the stretch of our muscles is really how we detect where our limbs are. And so that's the migration of the neural crest cells that are going to form the peripheral nervous system, is they're going to crawl just short distances on the different surfaces of the somites. The somites in the anterior posterior have different cell adhesion molecules. You want to be careful here because this is analogous to the segments in a fruit fly, not homologous. So it's different proteins that are setting up a boundary in every somite to make them express different cell adhesion molecules. So we just have to be careful not to say this is exactly like the fruit fly drawing assignment, but it's analogous to saying I like to crawl on elbow macaroni, and you'd be like, I'll crawl on your anterior segment cells because you're expressing that. And so that's cool because you can see that, right, our fruit fly story was we gotta make the teams, we're gonna make 2 teams. Once you've set up the 2 teams, other cells can use that as information and crawl across the surface of the cells. In a sense, we're saying that these neural crest cells are going to make the peripheral nervous system, they're going to make bundles of cells that'll be the dorsal root ganglia, and they do that by crawling over to the elbow macaroni team and saying, I'm not going to be on your team, I'm just going to be next to your team. Yeah? Okay? So that's that example. Woo. And then the next one, and our book has that nice figure, see, showing that. It's a complicated figure, but it's just showing that simple notion. Okay? And let's see, so the last one for neural crest cells is just that the neural crest cells also migrate to form our pigment cells. And so there's lots of different kinds of pigments in vertebrates. Think about a zebra. black stripes, white stripes, those are neural crest cells. Think about like a coral reef, and the fish on a coral reef are so colorful and so many patterns. And those are neural crest cells that started up here and take an epic journey out to the surface and have these patterns of different pigments in the cells. There's tremendous diversity of pigments in vertebrates. Birds, I guess those are feathers, so I don't know the deal with those, but certainly skin, upscales of fish are a story of neural crest cells like that. And zebras, and just lots of complicated patterns of pigments. So we won't dive into that, but just to have that awareness, because remember I said that neural crest cells are unique to vertebrates. And scientists think that maybe neural crest cells was the #1 innovation of the vertebrates in evolution, because it just provides so much potential, right, for a variety of different faces, birds, dogs, humans, and variety of different pigments. Like that. Yep. Okay. So just that. So we're good with that. First example, migrating cells. Neural crest cells form the vertebrate face, and vertebrate pigment cells, and vertebrate peripheral nervous system. They are crawling distances past other cells. Then, next examples, cells are holding hands, changing shape, sliding past each other. Our examples for that are our fruit fly assignment. The anterior-posterior assignment is a great example where the cells were not moving around, but the cell adhesion molecules are helping create a pattern. Then we have two more examples, and one is to go back to the Xenopus gastrulation, when we saw the belt narrow and fold in, and just review that and add this knowledge that the cells weren't really moving past each other, they were just shifting positions a little bit, and then add a new example, sea urchin gastrulation. So for this Xenopus gastrulation, this is what we saw before, where we start fertilized cell, then we have this rapid mitotic cleavage to make a hollow ball of cells, the blastula, and the cells that are fated to be ectoderm, mesoderm, and endoderm, they're all on the surface. We have a surface of ectodermal precursor cells on the animal poles, endodermal cells in the vegetal pole, and then this belt of mesoderm. Gastrulation is our example, where the cells fold in. And they do that not by migrating. They're not really crawling past each other. They're just shifting. And that example we had of that is, just take our class, and we're going to make it narrower, and making it narrower, it's going to have to be longer. And it's not going to involve anybody moving very far. Everybody just has to make it to the middle. So you're not really crawling very far, you're more jostling. And in doing so, it's just going to change the shape, okay? And we just have a couple of terms in that process. One of the terms is for the belt as it folds in. That's going to be a certain kind of cell shape changing. The other is the ectoderm that was up here is going to spread because it's the precursor for all of the skin. So it has to spread because of the embryo. And we haven't mentioned that movement before. But this is a great example because we're mostly familiar with it.
Is that going to be like through chemotaxis?
Is that like through chemotaxis? And the answer is yes. Yes. The book dives into that in some detail. The chemotaxis is always a story of potentially direct contact between a cell and other cells through cell adhesion molecules, or between a cell and the extracellular matrix through cell adhesion molecules. Or, that's direct contact, or there can be signaling factors that help it, right? So you could imagine that we want everybody to move toward the middle, and we just have a source cell, line of source cells in the middle, releasing a signaling factor, and that just says everybody get close to the signaling factor. Yep. And so this is a complicated image. It's just showing this idea of the belt changing its shape and folding in as it does. And the colors are just indicating what we talked about before. Oh yeah, you have this belt, but there is that belt buckle, which is red here. There's some pattern to the belt. You know, it's not just a simple leather belt, but there's actually some pattern because there are already some different fates along the length of the belt as it folds in. And this folding is an example of what's called in Xenopus, it's called convergent extension. And those are just two English words that are not really scientific words that are great, because we're going to have you guys converge to the middle of the classroom, and in doing so, we'll extend. So we're going to have the belt narrow by having the cells converge this way, and it will just naturally extend. So that in Xenopus is called convergent extension. That has a more general word because it's very, very common. And the general word is called mediolateral intercalation. And so that just looks like, cells in the classroom, and they're going to jostle together to make it narrower, and in doing so it gets longer. So they're going to converge and extend. And it's called mediolateral intercalation, and the word intercalation is also not a scientific word. It means to slide things past each other, like when you comb your hair, as your hair, the little tines on the comb are intercalating between your hair strands. Just means go between, slide between. So that's one example of very general. In the specific term in Xenopus is called convergent extension. The general term is called mediolateral intercalation. It's very, very common. Cells just slide past each other. The other example is what's happening. I don't really have a good image of this quite. The other example is here, where the blue cells are going to spread to cover the whole embryo. That's going to be the ectoderm. And they do that by, instead of having one room slide the other like this, imagine that the first, second, and third floor collapse. So we had students in 3 layers, or cells in 3 layers, collapse to 1 layer. And in doing so, we would just pancake out the whole classroom. So make it bigger classroom, because we just squeeze everybody together. You can see, oh, nobody's moving that far. We're just pushing it down together. And the general, or the specific term for that in Xenopus is called epiboly. And that's the ectoderm spreading. And there is a general term for epiboly. It's on this slide. The general term is the other kind of intercalation, and it's called radial intercalation. And here, the classrooms are squishing down, flattening it, and it just naturally spreads out. So that's what's happening. You say, look at the ectoderm spread to cover the whole embryo. What's happening? Just flattening down several layers to be 1 layer, and it naturally has to spread out. Yep. So let's pause for a little concept chat and talk about 3 things that are important so far today. And then we'll get the sea urchin example talked about probably. And we'll be ready for chapter 8, which if you came in late, we have only a set of pages we'll cover in chapter 8. And we'll talk about that on Thursday. Awesome. So find a partner and say, howdy. And then talk about 3 things that you thought have been important today.
I've just been writing yeah. And then we kind of and we talked about the long distance transformation of rivers, migration to form your vertical nervous system. Yeah. And then your pigmented stuff, which are like the stripes on a zebra or stuff. And then under that, we start with like skeletal stuff a little bit. After like, I accidentally commented on my seat. It was like sharp, I guess. Yeah, it's like a cough drop, but it doesn't have menthol. It's better for sickness and stuff. I know. Yeah. Dude, guess what? I turned 18 in 20 days. No, 30 days. December 19th. I can't wait. That's my golden birthday too. Because I was born on the 19th. What? This year? You're welcome. My sister's like she would like if she went to college, she would have graduated last year. And like it's weird to me that I think it's been so long. 24. I don't know, she turned 20. Oh no, I guess she's like.
Oh yeah. Okay, so Kim, what is one thing? Where's Kim? Hi Kim. What's one thing that your group thought was important today? Sorry, what? Yep, the dorsal root ganglia. So that is migrating. It's a good example because that's true migration. The cells are really moving past other cells, moving the same way as the neural crest cells move to form the face, or the other neural crest cells move to form the pigment. They're just not moving very far. Great. And from that, you can see it's really interesting, right, that when the neural tube folds, it folds up in vertebrates because of the signal from the notochord. And the cells that don't quite get included in the neural tube are the cells that become the neural crest. Because they're called neural crests because they're on the neural ridges, and the neural ridges fold up and they fold over and they make a tube. And the cells that don't quite get included in the tube become the neural crest. So you can see some kind of hints of the history of evolution where you're like, those cells, you know, the vertebrates took advantage of the cells that didn't quite get into the central nervous system, and they made other components of the organism. And they were, you know, migratory cells, so they had the potential to play, you know, you could migrate and do this, you know, job. And so there's a lot of potential for evolutionary innovation once you have a population of cells that are going to be migratory cells and they're ectodermal cells, like that, yeah? And so you see the neural crest cells make the peripheral nervous system a sense of touch and kinesthesia, you know, really closely related to the central nervous system cells. And then Kyla, what's the one thing that your group talked about?
We also talked about how the neural crest cells create pigment patterns in fish and zebra.
Yep. Yeah, and again, you know, classic tattoo opportunity, which would just be a bunch of arrows on your body, you know, like a lot of lines and arrows coming up, a lot of face tattoos with arrows zipping it up. That would be really cool, you know. And, you know, neural crest cells moving around. Lots of tattoos on your back, right? Dorsal root ganglia and stuff like that would be just epic. And final grade A. Come into the final exam with fresh tattoos, and I'll just be like, psh. And something I don't often remember to tell classes is I know how to go in and change all your grades, all the grades you've ever had. I'm just be like, raise them to A's. Yes, that's some nice tattoo work. All right, Mallory, what's one thing your group talked about?
One thing we talked about is just the 3 types of cell adhesion molecules, so the cadherins, CAMs, and integrins. But one thing I was curious about is like, because CAMs stands for cell adhesion molecules,
Yeah.
correct? I guess like my problem is like distinguishing like the difference of like why CAMs like falls in the category.
Between cams and cams.
Yeah,
that's my
so it's
question.
one of these just histories of, name issue.
Okay.
Like that, where, like I said, I don't know for sure, so I should be careful. But it's one of those things where they started naming things, and they're like, oh, we could call those cell adhesion molecules, and then you're like, that is such a great name. We'd like to use it as a general name. And that so a lot of times people don't even say CAM stands for cell adhesion molecule, because then people are like, I don't like that. But you see that every once in a while in science, where there's some specific name. And then like, oh my gosh, wish we could rename it, we could adjust that, and they just can't. A really famous example of that is Sonic Hedgehog. So the original gene was called hedgehog, and it was because in mutant fruit flies, they die at such an early stage, they just look like a little prickly hedgehog. So they named it that dumb name, and then they found the vertebrate ortholog, and it was one of the early orthologs where they're like, oh my God, we found this in vertebrates, but it's in vertebrates, so it's going to be way cooler. So we need a cooler name, and so they named it Sonic Hedgehog after the video game. And you're just like, shoot. And now people are really sorry because Sonic Hedgehog comes up a lot in genetic counseling. So like I have a friend who has a family, they run with extra digits. And Sonic Hedgehog plays a role in that. And so the doctors talk to him about the babies having an issue with Sonic Hedgehog. And the people are like, can you quit joking around? Because this is a real significant medical condition we're talking about. And the doctor's like, well,
Biologists are nutty.
but yeah, you can't rename it. You're just kind of stuck with it. And so kind of stuck with that, and so you try to slip through it. As a student, you try to slip through it without getting bogged down and too frustrated. But yeah, I mention it just because it's true. And sometimes if I don't mention it, people say, what's CAM stand for? And I'm like, well, yeah, but
You're
yeah.
not gonna like this.
Yeah, you're not gonna like this at all. Yeah, and that's one thing that scientists are not, in the path of discovery, no scientist is ever thinking, let's set this up so it's like, it will help future students. That's never part of the consideration. They're never thinking, yeah, why don't we just call them A, B, C, D? We're like, well, no, let's make them really funky. Like Zach is reading for his research article, a fruit fly article, and we talked about how they make dumb names for genes. So another dumb thing fruit fly geneticists do is they'll study a group of things. So he's studying an article where a hormone is synthesized, and it's synthesized by a series of enzymes, and each enzyme is a protein, so each enzyme must have a gene. And so the scientists that studied the genetic analysis of identifying the genes in that biosynthetic pathway said, let's name them by having a common name, and could be Halloween names. And so the genes names are like Phantom and Ghoul,
Yeah.
and just
Shadow.
just a series of half dozen names where they're like, ha 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 ha. Yeah, so that's not friendly, And but that's that. All right, so we have a couple minutes. Questions people have about anything at all? Yeah, Ali.
So do the neural crest cells actually make the skin and bones, or is it like the osteon?
So that colored part of the brain or skull, that front part of the skull in vertebrates is actually people say it's a derivative of neural crest cells. Yep. And people say, well, what's going on with that? That seems so contrary. It turns out that you could take a course that Rachel Simons teaches every other year, and it's called something like Comparative Vertebrate Anatomy.
Yeah.
And so it's about vertebrates, and it's about their anatomy. And if you look at the evolution of vertebrates, initially, early vertebrates were fish that, for example, had no jaw. So they didn't have a jaw, they just kind of, I think, had a mouth that could kind of go like that, just kind of close a little bit. And then another thing about them is they were covered with bony armor. And so they were just kind of cruising through the water without much flexibility, trying to protect themselves from big predators, and then just bumping into food. Not a winning strategy. But it's thought that the story of the neural crest and those bones is because there used to be a lot of ectodermal bones in vertebrates, because those bony plates were ectodermal, and they were bone. Remember that all cells have all the genes, so you have the potential for ectoderm to evolve the ability to form bones, and that all that bony plate armor was lost, except for up here. We have this bony plate armor up here that's now inside, like that. Yeah. So take Rachel's class. I don't remember if she's going to teach it again. I think she taught it last spring.
Yeah.
And it's fascinating because it brings a connection between these developmental stories and anatomy and vertebrate anatomy and the whole history of it. Yep. So we got to cut out of here. We'll talk just for the first couple of minutes about the sea urchins next time, and then chapter 8 stuff, differentiation and stem cells.
One of them is literally
Do you have time on possibly Friday for a meeting where we can show you working on a study guide, and you can just look at it and
Yeah.
be like, this is something we should know from this, or
Yeah, so
Okay.
I have to, my family is
I'm thinking we might have a-
celebrating Thanksgiving this weekend.
Okay.
And so I have to leave town at like about, and then I have a morning meeting, so
Okay.
should we try to plan a time right now?
Yeah, for Friday?
Yeah.
Yeah, because I was doing Thursday.